Everyone is being recorded, as if that's different than normal. So, my name is Ben Sykes, along with Helen Alexander. Welcome to the KU Ecology Seminar Series, which is sponsored by the Kansas Biological Survey. We're really glad that you're all here on this lovely Friday afternoon in the first week of October. I think, as you guys know, for those of you that have been here before, this is really for the new people, for people that haven't been to one of these before. Everyone is muted during the talks. We record the talks. If you have a question or an announcement that you want to make after the talks, just send it in the chat and it will come to me, and then we will call on people at the end. But otherwise, everyone is muted during the talk, okay? And we'll unmute individuals during the question and answer period. I think that's pretty much it. We might have one announcement about the email list, but we'll do that towards the end, okay? But we'll start with Naomi Betson, who's a PhD candidate in Brian Foster's lab, and we'll let Brian introduce her.
Great. Can you hear me okay? All right. Well, it's my pleasure to introduce Naomi Betson. Naomi is a PhD student in my lab. She came in 2018 after doing her bachelor's at Beloit College in the great state of Wisconsin. She's in her third year, and in a lot of questions in plant community ecology, particularly focused on restoration, invasive species, sort of spatial ecology, and also floral phenology, and those represent sort of components of her dissertation. And her dissertation work is focused really on two long-term experiments, one at the KU Field Station, which is a restoration study in tallgrass prairie, and that's what she's going to be talking about today. And I think it'll largely reflect what she presented in an ESA poster in August. The other experiment is a free state restoration project, which I understand she's not talking about today, but hopefully we'll hear something about that in the future. So with that, I'll turn it over to Naomi.
Okay, let's see if I can get the screen sharing to do its thing properly. Advanced portion of screen. Aha, and it looks like it's automatically mostly. Does that look okay to everybody? Yeah, cool. So, hi everybody. Thanks for zooming in today. I appreciate you joining me in this very odd week in what has been a series of very odd months. So today I'll be telling you about an aggregated seeding experiment out at the field station, presenting some of its initial results based on data from 2015, and introducing some next directions for the project. So why is spatial structure important for plants in the first place? Well, space is not only a key resource, but also a major niche axis that mediates plant interactions. It affects how plants use resources and what other plants they're able to interact with. And so this means it affects competition. Competition being vitally important for coexistence. So according to classic competition theory, species can coexist when they experience more intraspecific competition, that is, competition among members of the same species, than interspecific competition, which is competition between members of different species. This way they self-limit more than they limit others. And this requires interacting species to be sufficiently differentiated in niche requirements and resource use, while not being too different in fitness. And there has to be a sort of balance there. And so we know space influences these things. And so that's where spatial aggregation comes in. Intraspecific aggregation concentrates intraspecific interactions while limiting interspecific interactions. And so this may permit the coexistence of species that wouldn't normally be able to do so in close proximity due to strong niche overlap or strong fitness differences. Without spatial structure, if all of these species were to interact with each other equally, the dominant competitor would be predicted to exclude all of the other species with overlapping niches, while aggregation should cause the dominant competitor to self-limit. And aggregation also mimics the patchy structure that naturally develops in most plant communities due to seed dispersal, environmental heterogeneity, and a whole lot of other factors. And so what does all of this have to do with prairie restoration? Well, reconstructed tallgrass prairies tend to lack the biodiversity and floristic quality of remnant prairies. And this is largely due to a failure of seeded species to establish and persist, especially forbs, which make up most of the diversity in prairie remnants. Planting species uniformly throughout a site is common practice, but it may encourage competitive exclusion. In close proximity, the dominant competitors, here is usually C4 grasses, are able to exclude many of the less competitive forb species, and these are often the species with high conservation value. So we come to aggregated seeding. So there are certain pros and cons that aggregated seeding has over uniform seeding. In terms of benefits, it should reduce competitive exclusion of desired species, particularly by these dominant C4 prairie grasses, and thus improve biodiversity. On the other side, it might end up reducing competitive exclusion of invasive species as well. And then these invasive species could end up reducing biodiversity. And aggregation may also lower productivity due to less complete resource use, since these mixtures of species will use a much wider range of resources than monocultures. Previous research supports all of these to some extent, but has not examined which competitive interactions, those driven by fitness differences or those driven by niche differences, are most important in mediating spatial effects. So we come to the main questions of this project. Firstly, we want to look at how aggregation affects aspects of community structure. So how does interspecific aggregation affect initial prairie plant establishment, abundance, diversity, and floristic quality? And secondly, we address the mechanisms of the above effects. So to what extent are the effects of aggregation on the community the result of aggregating functionally dissimilar sown species, presumably minimizing niche overlap, versus aggregating functionally similar sown species, presumably minimizing fitness differences? And finally, to what extent does the aggregation of sown species influence invasibility by non-sown species, native and non-native? So the study site is a long-term aggregation experiment in tallgrass prairie restoration that is designed to test these ideas. It's located at the University of Kansas Field Station in a former agricultural field that was previously used for cool season hay, so species like brome and fescue. The site was herbicided and prepared for seeding in 2011 to remove all of these agricultural species and weeds. Then it was initially seeded 2011 and reseeded in 2012. I can show that, you can kind of see in this picture sort of one of the plots. You can see that they're pretty large. And so here is the experimental design. There are 25 of these 10 by 10 meter plots in a sort of 5 by 5 square. And so there are 5 plots for each of the 5 treatments. And within each one of these plots are 16 cells. And these are what allows for these different aggregation treatments that I'll be explaining. That means that there are 400 cells total, which means 400 cover surveys any year that this experiment is sampled. So that's it's quite a process. So these treatments are different arrangements of 16 species from 4 different functional groups. And that's really the only thing that's changed. So they're the same density in every single plot. So these treatments range from the uniform treatment, otherwise known as the uniform treatment, where all 16 species are in every single cell and they're all mixed up, all the way up to this aggregated monospecific treatment, or AgMon treatment, where each species is in its own cell. Then we have these intermediate treatments where there are 4 species per cell, and they have different levels of functional group aggregation. And so they range from the aggregated complementary, or AgCom treatment, where each species is from a different functional group, so they should be more different in niche and in fitness. Then there's the aggregated random treatment, where it's 4 randomly selected species. And then the aggregated redundant, or AgRed treatment, where they are all from the same functional group within each of these cells. And so they should be more similar in niche and fitness. And this is to test whether the effects of aggregation are due to separating species with strong fitness differences or separating species with strong niche overlap. And so here are the different species in the different functional groups. So we range from legumes are usually quite conservative and also not usually very high in fitness nitrogen fixtures, and then short forbs, which are also generally conservative and generally not super large or high in fitness. And then we have tall forbs, which are, as you can see, a lot taller than the short forbs, fairly self-explanatory name. And then grasses are C4 or warm season prairie grasses, which are often blamed for reducing forb diversity. And so these data are from 2015, the 5th year of the experiment. We're looking at results pretty early on in succession, so these results are likely to change over time. This data was collected before my time here, but had not previously been analyzed. So we come back to our 16 species again. We can see that though they were seeded at the same density, they end up varying very widely in abundance. And these percent cover values are from the entire experiment. So we can see that Sargastrum nutans ended up more than 400 times as abundant as Dahlia purpurea. Coreopsis lanceolata just failed to establish. But kind of the most important thing here is that the 6 most abundant species are all grasses or tall forbs. So there's definitely a difference in fitness between these functional groups versus the legumes and the short forbs. So question 1, our question of community structure. Well, we can see that the percent cover of sown and non-sown species across the different treatments, sown species end up decreasing with aggregation, and then non-sown species increase. And this kind of shows evidence of self-limitation of sown species when they're aggregated. And then here we show a bunch of different values of community, a bunch of different results related to community structure, such as richness, the overall richness of all the species, sown and non-sown, then broken down by sown and non-sown, as well as the evenness of sown species and abundance weighted floristic quality index, which floristic quality is a measurement that's based on coefficients of conservatism, which are measurements of how exclusive species are to higher quality habitats. And so richness increases with aggregation, and that's specifically due to these non-sown species. The sown species are fairly constant in richness across the different treatments. Evenness of sown species also increases with aggregation. And then the abundance weighted floristic quality decreases with aggregation. And we did look at other diversity indices for the whole community, but richness was the only one that was significant. So how does interspecific aggregation affect initial prairie plant establishment, abundance, diversity, and floristic quality? Well, aggregation increased overall species richness due to increased richness of the non-sown native prairie species rather than an increase in sown species. And then it also decreased abundance weighted floristic quality due to a decrease in the abundance of more conservative sown species and increase in the abundance of the more weedy non-sown species. And these results are more or less consistent with expectations that aggregation would cause the competitively dominant species to self-limit, improving coexistence. In this case, it's coexistence with non-sown species rather than with the less competitive sown species. And so we come to question 2, where we look at the mechanisms behind these effects. I bring this up again just to show that in these intermediate treatments, there really isn't much of a difference for most of these responses between the 3 different intermediate functional aggregation treatments. There is for floristic quality, but not for richness or sown evenness or non-sown richness. So what do we see instead, and what's going on with these functional groups? Well, the functional groups respond pretty dramatically in terms of they have very different responses. The percent cover of grasses and tall forbs decreased with aggregation, while short forbs and legumes were unaffected. Grasses were more suppressed by members of their own functional group, while tall forbs were relatively equally suppressed by all different species. So there's definitely some interesting effects there that are specific to these different functional groups. And here we show the relationship between the effect size of aggregation and species abundance rank. So this aggregation effect size is this log response ratio of cover in the monospecific treatment to cover in the uniform treatment. So it kind of shows how different this monospecific treatment is from the uniform treatment. And the lines represent 95% confidence intervals. So effects are significant when they don't cross this 0 line. And so we can definitely see that the effects of interspecific aggregation varies among sown species as a function of their abundance in the entire experiment. And that the most abundant species are most strongly inhibited by aggregation. And these are mostly our sort of 2 most abundant grasses, our 2 most abundant tall forbs, and our 1 legume that is even very abundant at all. So to what extent are the effects of aggregation on the community the result of aggregating functionally dissimilar sown species versus aggregating functionally similar sown species? Well, as these effects were largely reflective of non-zone species, they were more led by specific functional groups than by the arrangement of these functional groups. For metrics such as richness in zone species evenness, it really doesn't matter so much whether you separate similar species with similar niches or species with similar fitnesses. So the two most abundant zone functional groups, grasses and tall forbs, were both abundant enough to spatially self-limit when aggregated, as well as being responsible for suppressing the non-zone species. Those responses that did differ among the functional aggregation treatments showed that the aggregated complementary treatment was more similar to the uniform treatment, while the aggregated redundant treatment was more similar to the aggregated monospecific treatment. So this kind of means that these effects of aggregation are largely due to aggregating species of the same functional group and separating species of different functional groups. So separating species with strongly different fitnesses has more effect on those responses that did end up differing, which are the zone species abundance, the abundance of grasses, and then the floristic quality index. And so now on to question 3, our question of invasibility. We bring this back up to show the decrease in zone species, which allowed a sort of invasion of non-zone species. But then once we break down this non-zone species response, we see that it's completely due to these non-zone native species, since non-zone non-native species were very low in abundance and there was really no significant effect. So to what extent does the effective aggregation of zone species influence invasibility by non-zone species? Well, though aggregation suppressed the abundant zone species, leading to increased invasibility by non-seeded native species, there was no corresponding increase in non-native species. This may simply be due to the low abundance of invasive species, and many of these native non-zone species weren't exactly desirable prairie species. So we had a lot of ragweed, and then we also had dogbane or avocynum, but then we also had some reasonably conservative species, such as Pycnanthemum tenuifolium or slender-leaved mountain mint, shown here with the butterfly. And this effect is likely going to be specific on what site you're at. So the composition of the surrounding area is going to have a lot of effect on what potentially could enter your system. So here are some conclusions. Firstly, we can safely say that intraspecific aggregation strongly affected plant community structure in the early establishment phases of restoration. And these results were largely consistent with the predictions of spatial competition theory. Aggregation reduced the total cover of zone prairie species due to strong self-limitation of the most abundant and productive, but then contrary to our expectation that the less abundant zone species would be positively affected, you can see that the less abundant zone prairie species were unaffected, and this is possibly due to suppression by non-zone species in the more aggregated treatments. Intraspecific aggregation increased richness due to an increase in non-zone species richness and increased evenness among zone species. And these were both due to a decrease in the abundance of zone species. Aggregation also increased community invasibility by non-zone species, but these were primarily opportunistic native species of low conservation value. So some takeaways and implications for restoration are that spatially aggregated seeding may initially improve biodiversity in restorations, but at the cost of floristic quality, as it suppresses abundant zone species while promoting the establishment and persistence of non-zone species. This means that the benefit of aggregated seeding as a tool to improve restorations is dependent on the local species pool. If you're right next to another restoration or even a remnant prairie, then you might want to aggregate and allow in a lot of non-zone species. If you're near a patch of invasives or some real brambly woodsy area, then aggregation might not be the best idea. And of course, these results here show very early dynamics, and there have been major changes since 2015, so the long-term outcome may be different. And speaking of that long-term outcome, the future directions for this project will be comparing this initial data from 2013 through 2015 to newly collected data from 2019 and 2020 to see how the patterns develop over time and whether any new patterns emerge. So we have a sort of very preliminary result here that's from data we used from 2020, and it shows a very similar pattern of zone and non-zone abundance, but a much lower abundance of these non-zone species overall. So we can see that there's been a sort of development in this system that's trending more towards these zone species, these zone prairie species. And so I'm looking forward to seeing how the rest of these results have changed. So we got our acknowledgements of funding sources, lab assistants, undergraduate assistants, many, many, many people who have helped out with this very large project. And any questions?
Yay, well done. So a couple of people have sent messages, so I'll just call them, Naomi, and then we'll let them unmute
Okay.
and they can ask their questions. Okay. So how about Rob? You had sent me one first. Go ahead. Or actually, Helen's got an announcement real quick. Rob, why don't we let her go first with her announcement and question, and then you can go second, okay?
Yep. Well, why don't I do the announcement and then I can do my question in order. I don't want to jump in, but I did want to just remind people that we have this semester pretty well signed up, but due to a few changes, October 23rd is now a slot that is completely open. So if anybody's interested in giving either a short or full talk, let me know. And the spring semester is also fully open. But I'll wait on my question till others ahead of me had a chance.
Cool. Go for it, Rob.
Hey, Naomi, fantastic talk. It was really interesting. I don't know if I fully understood one part of it, so I just want to ask. So are these aggregated, like the tall grasses, or the grasses and tall forbs, are they experiencing more negative impacts from aggregation than they would in a pure monoculture, do you think?
Yeah, because you can, well, then in a monoculture. That's actually, yeah, because aggregation is kind of, yep. I guess, that's a bit of a tricky one. It's a bit of a tricky one to think about because it's kind of similar to monoculture, but then there's an allowance for these non-seeded species to come in. So it might end up being more negative overall, I guess, depending on what non-seeded species are there. It would be hard to say without testing. So yeah, I'm not entirely certain for that one.
It was just a thought I had. It's just really interesting overall.
No, that is an interesting thing to think about.
Thank you.
Thank you.
So I think I'm next. So I just love this experiment. Thank you for sharing it with us. So I was trying to look at all those species and obviously couldn't keep track of it, but
oh yeah, I can.
Well, no, I didn't expect to. What I'm trying to think about is they vary, I think, in how much they have kind of clonal spread versus really dependent on seed. And maybe that wouldn't have much effect early in the experiment, but I would think as the experiment progressed, it might. And I haven't really thought this through, but I was just curious if you had any thoughts on that.
That's also a good point. Yeah, it seems like that would definitely have some effects overall. I mean, it might be, even early on, it might be contributing to some of the difference in overall abundance, because those are some pretty spectacular differences. And it certainly would allow the species to grow in more close clumps naturally, so they might be more used to aggregation. Not sure exactly what effects those would have, but
yeah, I think it's just interesting thinking how these different species spread, because some spread clonally, some may produce just a ton of seed but disperse at fairly large distances. Anyway, I haven't thought it through either, but I think it would be a neat part of the biology to try to think about if you can.
Could definitely affect how quickly the species could spread into sort of areas that are left open. Might affect that.
Yeah, traits related to that spread are really important, or could be, yeah. Jude, I think you were next.
All righty. Thanks. Always impressive to see what's going on out at the field station. For somebody who sits behind a computer all day, doesn't get his hands dirty. Well, I'm not an ecologist. My questions are probably more answerable than the other two. the first one being, so I noticed your percent cover values added up over 100. Are you
Oh,
using
yeah.
area index or what's the explanation there?
That's because overlapping canopies. So if there's like layers and layers, so you can have, the ground being entirely covered by one species and then other species that are growing up over them with large leaves. So it ends up with, just because species being very tall, so we can have values over 100%.
Okay. I mean, did you use something like a leaf area index? Or?
It's eyeballed. Honestly, that's you can't 400. These are, it's again, there's 400 cells to survey, so.
Understood. And so now that explains Brian's backdrop too, after seeing the picture in your talk.
Yep.
So did you have, is that from, is there a camera on a pole or overhead? Like,
oh, that was, there were some people from the sort of advertising department that came to take, I think you can find it on like the main college website sometimes when you just go to the ku.edu, it'll have like drone, because they used a drone to get that picture.
Excellent.
Yeah.
Let's see, one of the other questions, trying to think back, was, so when you were showing the ratio with the natural log function.
Oh, yeah.
And the regression, what was that? So I look at those points and yeah, I mean, they're kind of linear, but they kind of lean on the ends. Is that an equation that you already, that's already sort of was determined beforehand, or did you land on that relationship after?
No, it was determined beforehand. So, and you can see it's a pretty decent fit here with the R squared value. But no, it was, I guess, yeah, we decided to use this log response ratio beforehand.
Okay. And I guess the final question would be, is there any, can you characterize then I mean, well, I guess you already have somewhat in as far as native or non-native, the non-sown species that ended up moving in. Did you see any sort of trends or patterns with those?
Not sure exactly what you mean there.
Yeah, like I don't know, particularly dominant. I mean, you mentioned ragweed and things like that. Was it that kind of really opportunistic stuff that was the bulk of the invasive?
It was mostly sort of agricultural weeds. They're probably just lurking around since it was in former agricultural land, kind of waiting for an opportunity to jump in.
Okay.
So if you look around there right now, there's a lot of Apocynon cannabinum or dogbane that's just like on practically forest of it sitting right outside the experiment at the moment. So it's a lot of things
Dogbane's
like
been acting crazy. Dogbane sometimes acts a bit invasive, I feel like, or at least weedy, certainly.
You look at it out there and it's a little scary looking.
Yeah. I was gonna, Jude, were y'all done? Sorry, wait, I was gonna ask a question for Wim.
This very last quick one is, and so this plot is it pretty much been, Ronco set it and forget it as far as management?
Oh, there are burns that are done. So we're not removing invasive species because we want to test invasibility. So we can't really remove the invasives, but there have been controlled burns every year. So.
Okay, thanks.
So that was part of Weba's question I was going to answer or ask for her was about the disturbance regime there. But her other question was about whether or not the original restoration was done by drill seeding or by broadcast, how you guys actually got the treatments imposed.
I think broadcast, but I have to look up because I was not there, so I forget specifically.
It was hand broadcast.
Yeah.
Hand broadcast.
And you said it's been burned, Naomi. Do you know the frequency or anything else, anything more in depth about every year it gets burned?
Every year it was burned, yeah.
Interesting, given how you have a lot of really tall plants. Kind of, I was thinking that must be because it hasn't been.
Oh no, they'd come back.
Yeah, they are.
Well, they're not as tall as they are out at like Free State,
okay.
Which is another prairie I go to. So the species out at this experiment, or even the tall ones, don't seem to get quite as mind-bogglingly, 6 foot tall as they do somewhere else.
So I'll, if you can stop sharing your screen, I'm going to let Lígia get going, but Camille might have a question for you, Naomi, that she can slip in as Lígia is starting her screen up.
And
Hi. I was wondering how, maybe I missed it, but how did you actually determine floristic quality? Is it like lots of different variables that determine that, or is it just the score? Or like,
yeah, it's the coefficients of conservatism and then the relative abundance of these different species. So it's kind of the average weighted for abundance. It's the average of these coefficients weighted for abundance. So it's really there's one of these values assigned for all the different species. We stick them into an equation that features the abundance, and out pops the floristic quality.
okay, yeah, I was just wondering if there were variables you might use, but thanks.
No,
Cool.
it's really just these coefficients and then how abundant all the different species are.
So thank you so much, Naomi, and other people can ask questions maybe offline. We're going to go ahead now and pivot to our second speaker, Lígia Souza, who's going to be introduced by Sharon Billings.
Hi, everyone. So I'm happy to introduce Lígia Souza. She comes to us from the University of Viçosa in Brazil, where she received a master's degree in geography. And she was studying a lot to do with soils. She was looking at erosion and different soil characteristics and remote sensing data that can help us understand erosion patterns. She is working towards her PhD here, transforming some of her soils and geography knowledge into ecosystem ecology. She's got quite a bit of skill sets. She has been able to take a course in global carbon cycle modeling at Northern Arizona University as a student here at KU. She also has participated in a neon workshop in Colorado to learn how to deal with neon data specifically. And to complement these skills in really large data sets, she's quite adept in R programming. So here in my lab, she's been working on the EPSCoR project, looking at stable isotopic signatures and radioactive radiocarbon signatures of soils, and attempting to understand how climate and rooting abundances that change with land use will affect carbon and nutrient cycling in the future. So she is very adept at thinking about how really small scale processes in ecosystems linking roots and soils can influence very large scale patterns. And I think that's what she's going to be speaking with us about today. So I'll let Lígia take it away.
Well, thank you all for being here today virtually. So today, if you know me, you've probably seen me talk about enzymes before, but today I want to talk about this approach to understand how microbes may be perceiving their environment by using soil enzymes. So, and why soil enzymes? So enzymes in the soil are often used as a proxy to understand organic matter decomposition. And that's because they can catalyze reactions in the soil that transform complex organic molecules into simpler forms that are readily available for uptake. So for example, a simple phosphomonoester here with a phosphate requires a hydrolysis reaction by an enzyme to release that phosphate. So enzymes then are specific to a type of chemical reaction in substrate, and they have several sources in the soil as they can be synthesized by microbes, plants, and animals. And as soil organic matter decomposition is strongly dependent on microbes and enzymes, these are commonly used to understand the soil energy and nutrient status based on the roles that they play in these processes, as we can see here in this table by Yang et al. That shows some of the enzymes in the soil and the functions that they that they play. So the reaction I showed, the release of phosphate, is then possible by the activity of phosphatases, which are enzymes commonly used to understand the phosphorus status in the soil. So, but the enzyme activity is dependent on some factors such as soil texture, organic matter content, and soil pH. And different enzymes show different pH optimum because the pH can affect the conformation of the enzyme in the soil, the absorption of the enzyme onto soil surfaces, and the ionization and solubility of substrates that affects the substrate diffusion in the soil. So in this study from 2010 by Turner, studying tropical soils, they showed how commonly characterized enzymes here, beta-glucosidase, which I'll be calling BGAs, and it's responsible for the release of carbon in the soil, and chitinase that I'll be calling nagase from now on, and it's responsible for the release of nitrogen in the soil. So this study showed how the activity of the enzyme here in the y-axis is dependent on the buffer pH. And here the buffer pH is when we perform enzyme assays in the lab, we make a soil slurry, and the slurry uses this buffer that has a preset pH. And what Turner found is that the activity of the enzyme is at its highest closer to the pH optimum of the enzyme, here around 4 and 5 for both enzymes. And this is important because some protocols suggest that we run enzyme assays at the optimum pH of the enzyme. But if the environmental pH doesn't match that optimum pH, we may actually be overestimating the activity of the enzyme in the soil. And a previous study here from our lab, the Billings lab here, studied these enzymes, so beta-glucosidase and chitinase nagase, to understand changes in the release of carbon and nitrogen. And here we can see in the y-axis the carbon to nitrogen release ratio, and we can see how it changes with temperature here, represented by the different points. But the magnitude of that change is dependent on the soil pH here. So we can see as we move along the gradient, the pH gradient here, that magnitude of response will change significantly. Similarly, I'm working on doing the same, but for acid phosphatase. So I'm trying to understand the kinetics of acid phosphatase by also using purified enzymes. So here, this is a study using purified enzymes to understand the release rates. This here uses soil samples, the enzymes from the soil. And here with the acid phosphatase, I'm also doing it with a purified enzyme to understand here the rates of the activity here displayed in the y-axis, how they respond to different temperatures here shown by the different colors. Here the temperature, and we see how pH mediates the response of the enzyme. And this is a nice way for me to talk about phosphatases, which are a unique type of enzyme, because unlike most of other enzymes, phosphatases can be divided into two major types, so an acid and an alkaline phosphatase. And this division is based on the optimum pH of each enzyme. So acid phosphatase is commonly predominant in more acidic soils, and alkaline phosphatase is predominant in less acidic soils, so neutral to alkaline soils. However, it's important to note here that the existence of both enzymes in the soils are not exclusive, so they both coexist in the soil. So in that same study from 2010 by Turner here, studying the phosphatases, we can see the activity here on the Y, and we see the buffer pH 2 here in the x-axis, and what we see is two different peaks of activity, and here we can see peaks closer to the more acidic optimum here, so characterizing the acid phosphatase predominance, and here we have the alkaline phosphatase more predominant. So, and it displays really well this duality of phosphomonoesterases, so phosphatases, that have these two pH optimum. And with that in mind, a study from 2000 proposed to use a ratio of alkaline to acid phosphatase. So here in the ratio represented the alkaline by one of the graphs from Turner with the peak around the optimum pH for alkaline phosphatase, and here with the optimum for acid. And so they hypothesized that an adequate level of pH for crop growth would be reflective of a proper ratio of alkaline to acid phosphatase. So they did that for agricultural soils. And by doing that, the authors suggested that this might be a more accurate approach to assess effective pH, as enzymes can integrate the soil chemical, physical, and mineralogical parameters into a single response. So this is a really nice way to ask the microbes how they're perceiving their environment instead of measuring it by using large electrodes, as we commonly do to measure pH. So then we had this, we posed this question if this ratio of alkaline to acid phosphatase is congruent with the soil pH across land uses, because that paper from 2000 used it for liming purposes in agricultural soils. And we were wondering if we can use that to assess the effective pH across land uses. And we also wanted to see if we can use this to evaluate the soil microbiome's response to these changes in land use. So to do that, we use soil samples from different soil horizons from 3 different land uses. And here I want to point out that we use the soil's genetic horizons and not by fixed depth. And though they have unique genetic horizons, I'll be calling them as horizon 1, 2, and so forth. And we then got these soil samples from 3 different land uses for native, for agriculture, and post-agriculture. And we measure the potential enzyme activity of both enzymes to calculate the ratio. And here I just want to point out that the continental US shows this really nice gradient of pH from coast to coast, where we can see more acidic pHs represented in red colors and more alkaline pHs represented in blue colors. And the reason why I bring this up is because Kansas is a good representative of that pH. And here in the stars, we are showing the places that we sampled. And you can see that we have this representation of a nice pH gradient from west to east, but also we have this pH gradient as we move deep in the soil. So here we have surface soils, and here we have deeper soils, and we can see that the bluer, there are more bluer colors here deep in the soil. So our preliminary results here are shown in the graph with the soil pH here in the x-axis, and we have the log of the ratio here on the y, and we can think of more negative results showing the greater relative activity of acid phosphatase activity. And what we see here is that in the native site, we have a significant relationship between the pH and the ratios. And here the points represent the horizons, as I said in the previous slide. So as presented here sequentially. So lighter colors represent more superficial horizons, and deeper soils are represented by darker colors. Again, so here we see that the relationship is significant, and we also see the significance under post-ag sites, which is a good indication that there might be hope for restoration because both of these are similar. But this relationship falters under agriculture, as we can see here, the p-value of 0.55 that displays no significance between the ratios and the pH under agriculture. But a nice feature here is that we can almost see a division between surface and deep soil horizons. So we decided to analyze them separately. And what we found, again, here with the pH on the x and the ratio on the y, is that this relationship between the pH and the ratios is even stronger in surface horizons, though it's still not significant under agriculture. But though it's interesting to note that the p-value here decreased in comparison when we use all the soil horizons. And it's also interesting to think about this p-value when we consider only the deeper horizons, because then again, soil pH here on the x and with the ratio on the y, we see that the relationship between the ratios and the pH falters under all land uses. And interestingly, under agriculture, we see a decrease in the p-value as well. So we hypothesized that because this ratio doesn't correspond to the pH at depth, we hypothesized that the microbiome at depth might be somewhat disconnected from the surface. So changes to the land at surface are not being reached, are not reaching down deep in the soil. And this small value here for samples from agricultural sites may be an indication that the changes in land use are actually allowing this communication between these two worlds, the surface and the deep soils. And one of the reasons that could be happening is because, for example, soil structure is being altered under agriculture. As we know, for these sites, we have greater proportion of macropores, for example, that can allow this communication. So, but why is this significant? So here I have a graph of soil pH here on the X and the depth of the sample in the Y. And we see here that the land uses, we see here the responses for each land use. So the red points here represent the agricultural sites, and the green and the orange represent the native and the post-ag sites respectively. And we see that the pH has changed under agriculture, more so at surface than at depth, where it's quite similar across land uses. And if we compare this to the alkaline to the acid phosphatase ratio, what we see here in the x-axis is the ratio, and again the depth on the y. What we see is that the trends are similar at surface, although not as similar for agriculture as we would expect, as we've seen in the results in the previous slides. But it's somewhat similar at surface, but then at depth, the trends are reversed. So we see a greater relative activity of acid phosphatase at depth, but the pH is actually increasing, so the result is not as congruent. So this could indicate that changes at the soil microscale may be occurring that are not being captured by standard approaches, and could even be occurring at shorter time scales than previously thought. And if this is true, it could have important implications for energy and nutrient cycles, as they can affect the release from organic compounds by enzymes, which is a nice circle back. And here, so in our work with the purified acid phosphatase, we were able to demonstrate the importance of pH for the release of these organically bound phosphorus by these enzymes. And here, this graph shows how the temperature sensitivity of the acid phosphatase, represented by the activation energy in kilojoule per mole, changes as we move across the pH gradient, here represented in the x-axis. And this gray area here corresponds to the standard deviation of the curve. So this can have implications for our understanding of how phosphorus availability may change under warming, and how, depending on the soil pH, increases in temperature may not promote greater releases of phosphorus, in this case, from these organically bound sources, because enzyme activity may not be responsive to temperature. So thinking about this spatially explicit, so let's think about the graph that we've seen in the previous slide, and this is the spatially explicit version of that. And what we're seeing here is the temperature sensitivity again, as the activation energy in kilojoule per mole. As we have darker colors, we have more sensitivity of the enzyme to temperature. So warming would increase the release rate of phosphorus in these places with darker colors here. But if we keep changing the landscape as much to alter the soil pH, this can affect the degree to which the release rates of organically bound nutrients, such as phosphorus in this case, are going to respond to warming. So this has really important implications for global cycles, global nutrient cycles, and our predictions of how the world is going to look like under warming. So preliminary conclusions from this this study using the phosphatase ratios is that they do show potential to be used as a proxy to evaluate changes at the soil microscale. And also, preliminary results suggest that land use affects the communication between the soil microbiome at surface and at depth in a way that maybe is not captured by conventional methods, such as a chemical approach to soil pH. And with our next steps, we want to analyze the phosphatase ratios in combination with other data sets, such as the root abundance and the soil aggregate size distribution, so we can understand, especially at depth, why is that change occurring? Why is an increase in pH is not being followed by an increase in the ratio of alkaline phosphatase ratios? Lots of people to thank here. I want to give a big shout out to my lab group for all the fun science conversations. Theo also for helping me out a lot and talking to me about all stuff science, which is really cool. Regina, who helped me with the essays in the lab. The KU Field Station for the award and allowing me to pursue this acid phosphatase route and the ways in we can use it to understand land use legacies. The Kirk Lab at K-State for providing the pH data. The Kansas NSF score for funding, and also all the team that has helped with the collection of samples and the homogenization and all the process, and the NRCS for digging up the bits for us. And thank you so much for being here, and I will take any questions, comments, and feedbacks you may have for me at this moment.
Hooray! Okay, so if you have a question, please do send a message in the chat. It looks like Camille has one, and she can be up first. Make it where participants can unmute themselves. Go for it, Camille.
Hey, that was awesome. I had a question with the last global map that you showed.
Sure.
Because it depends on land use. Are you thinking about like overlaying land use?
Yes.
With temperature to see how like the intensity of land use might alter stuff?
Yes, so for the phosphatase, actually, it's not as responsive to temperature as we might expect, but it's really responsive to pH, as we could see in that graph. So I'm interested in overlaying land use, so I can try to understand like areas where changes in pH might change that response, and then the release rate of the phosphorus.
Cool, thanks.
Yeah.
Cool. Jude has a question or two. Maybe be two max, Jude. Are you there? I have one if not.
Okay. All right. Sorry. Oh, now I got to move my microphone. Yeah, and thanks with this map up too. I mean, it looks like we're almost looking at a forest map, forest percent cover map.